Hello to all. Welcome back to another session. Today we are talking about detailed structure of microsporangium and microsporogenesis. So microsporangium. So we have already discussed in the previous class that microsporangium, that is the singular form. The plural form, that is microsporangia. So microsporangia, this word, this microsporangia, it is basically used in the young anther. So in young anther, microsporangia are formed, and later on this microsporangia, it forms the pollen sacs or pollen chamber. Okay, pollen chamber. This is one microsporangium of young anther, and this microsporangium, it further develop to pollen chamber of mature anther. Mature anther means here the pollen grains are formed which ready to released. So this microsporangium, it is more or less circular in nature and it's surrounded by four layers. Okay, this is surrounded by four layers. The outer layer, there is epidermis. Next layer, there is endothecium. Next, middle layer, and next tapetum. Okay, epidermis, endothecium, middle layer, and tapetum. So these are the four layers which forms the outer boundary of microsporangium in case of young anther. So out of this four layer, epidermis, endothecium, middle layer, and tapetum, this three layer, okay, except tapetum, this three layer, it forms or it functions as protection and dehiscent of anther to release the pollens. Okay, this three layer. Next, the tapetum. The tapetum, it functions as nourishment due to the presence of dense cytoplasm, and this tapetum, it consists of more than one nucleus. Here, this layer, this tapetum, it consists of more than one nucleus and dense cytoplasm. It is as compared to the other cell, you can already see that this is very dense. The cytoplasm is very dense, which consists of more than one nucleus. Means this tapetum is multinucleated. Here the question is there, that is, can you think of how tapetal cells could be binucleate? Okay, as you know that this tapetum, it consists of dense cytoplasm and having more than one nucleus. How? Because these tapetal cells are this cell. Here it is. Okay, this cell, it is very large. Okay, it is very large with more than one nucleus, and they assist with pollen grain growth and development. Means nourish this developing pollen grain. What is the function of this tapetum? It nourishes the developing pollen grain. And this tapetal cell, it undergo endomitosis, where the nucleus in the nuclear membrane splits, but cytokinesis does not occur. Means division of this nucleus takes place, not followed by the division of the cytoplasm, so that in one cell many nuclei are formed. You can also say that here the chromosome of, or this duplication of this chromosome occurs, that result in the formation of a more than one nucleus. Okay, so they become bi or multinucleate because of only these. So this microsporangia, it's surrounded by four layer, and this is the most innermost layer, that is the tapetum. Okay, which functions as nutrition, provide nutrition. Next, the fertile part of this microsporangia is known as microspore mother cell or sporogenous tissue. First, sporogenous tissue is developed, and this sporogenous tissue, which are compactly arranged, these are compactly arranged. This sporogenous tissue, it further divided to form the microspore mother cell. Microspore mother cell, it forms from sporogenous tissue by mitotic division. This is the sporogenous tissue. It further divided mitotically to form the microspore mother cell. Okay. This sporogenous tissue, it is homogeneous, compactly arranged cell. Homogeneous means all these cells are same. and this present in the center of this microsporangium. Sporogenous tissue, it mainly occupy the center of the microsporangium. Clear? Next, we are going to discuss about the microsporogenesis. And remember one thing in exam here, the questions mostly comes from this topic. Means here is means what is the function of this tapetum? What is the function of all these three layer? Okay, why it is binucleate? Clear? Next, microsporogenesis, microspore, and genesis. Microsporogenesis, genesis means formation. So the process of formation of this microspore or pollen grain is known as microsporogenesis. So as this anther develop, means after the anther it get mature, then the cells of the sporogenous tissue, it forms the microspore mother cell, which undergo meiotic division to form microspore tetrad. The microsporogenesis, the process of formation of this male gamete or microspore, formation of this microspore. So this sporogenous tissue, also known as pollen mother cell, it divided to form the pollen mother cell, which is also known as microspore mother cell, which is also known as potential pollen. What is potential pollen? It means each of this cell, it has potential to form the pollen grain. That's why this mother cell is also known as potential pollen, which is diploid in nature, 2n. Okay. So this microspore mother cell, it undergo meiotic division to form the pollen grain or microspores. Okay. Microspore tetrad. Why it is known as tetrad? You know that if this cell undergo meiotic division, it forms 4 haploid cell. The cell itself diploid in nature, always diploid in nature. That's why it undergo meiotic division to form haploid cell. Okay, so how many haploid cells are produced? 4 haploid cells are produced. That is known as tetrad. Here is tetrad. This tetrad, it arranged in linear manner. It also arranged in this manner. Okay, tetrahedral manner. So how many pollen grains are produced from one pollen mother cell? There are 4 pollen grains are produced. Here the question is that what would be the ploidy of the cells of this tetrad? That is haploid. Here this is haploid. N means haploid. So here this sporogenous tissue, it gives rise to microspore tetrad, and it is also known as potential pollen or microspore mother cell. And this process of this formation of this microspore from microspore mother cell or pollen mother cell, also known as microsporogenesis, which are arranged in tetrad. That's why it is known as microspore tetrad. Okay. After the formation of this microspore, they get dissociate or separate from each other to form free pollen grains. Okay. So here, this is the structure of this mature anther where already pollen grains are formed, which are haploid in nature. And in mature pollen grains, here in this mature anther or in this one microsporangium, it forms the pollen chamber. And in one pollen chamber, several thousands of microspores or thousands of pollen grains are formed that are released with descent of anther. Okay, how they get released? They release from this line of descent. So this is the line of descent. Okay, or this is also known as longitudinal groove. This is the longitudinal. So look at this. These two are the longitudinal groove where after the pollen are formed, these pollen grains, they get released in this side, not in this side. Okay, not in this side, they get released in these two sides. That's why this is known as longitudinal groove or line of descent. This is the line of descent where the anthers are open. Okay. So if 1,000 of pollen grain mother cells are produced in one microsporangia, then 4,000 microspores are produced, or 4,000 pollen grains are produced in each chamber. So this is all about the microsporogenesis. Please remember, and this diagram is very much important for exam point of view. So please practice this diagram. Clear? Thank you.